Good morning, everyone. Welcome to the fourth-day session of the Faculty Development Program. Myself, Dr. K. Swati Priya, co-convener of the Faculty Development Program conducted by Srinivasarao College of Pharmacy on the theme Challenges and Innovations in Advanced Pharmacognosy. Our, Now, I request Mrs. Dr. D. Nivedita Ma'am, HOD of PharmaD, to introduce our today's resource person.
Thank you, Swati Priyama. A very good morning to one and all. Myself, Dr. D. Nivedita, Assistant Professor of Pharmacy Department, Srinivasarao College of Pharmacy. Take immense pleasure to introduce our today's guest speaker, Dr. Gummadi Vedapriya ma'am, to the session. Dr. Gummadi Vedapriya ma'am works as Associate Professor in the Department of Pharmacognosy and Phytochemistry in the Aditya College of Pharmacy. Madam has completed B Pharmacy in Avanti Institute of Technology in the year 2009 and completed M Pharmacy in Andhra University in the year 2011 and completed PhD in Pharmaceutical Sciences in the Andhra University in the year 2018. Dr. Gummadi Vedapriya ma'am loves teaching, and with the teaching experience of over 10 years, she has guided 15 UG projects and 6 PG projects and turned many challenges to triumphs. Madam has more than 15 publications, in which 5 are Scopus, 3 are UGC, and 3 are of Web of Science. And Madam is very interested in learning and updating her knowledge, so she has attended more than FDPs, 20 FDPs, and seminars. And Madam has a membership in the APTI and Pharmacy Teachers Trust. Thanks a lot, ma'am, for being here with us today. And I request Dr. Vedapriya, ma'am, to take over the session. Thank you, ma'am.
Thank you, Dr. Nivedita ma'am, for your nice introduction. And today I would like to discuss about pharmacognosy, myths and challenges in the 21st century. Hope I'm audible to everybody. Next slide. So today, these are the contents. Today, I will be discussing about pharmacognosy in the 21st century, acquisition of medicinal knowledge in the past, modernization of pharmacognosy, challenges for developing standardized herbal drugs of population, need of quality assurance, and herbal drug opportunities. Next slide. So, pharmacognosy in the 21st century, as we all know, there are a lot of development and advancements in the 21st century. So, in the last few decades, there has been an increasing use of biogenic resources when compared to previous decades. At present, it must be stated that general interest in pharmacognosy is very high. Simultaneously, more people are trusting in medicinal products. In some countries, phytopharmaceuticals, also known as phytotherapy, are still used as an integral part, and interest in these compounds of natural remedies is also growing in the scientific community. There's increasing scientific research in the components of natural medicines and nutraceuticals, increasing funding opportunities, but also increasing competition from those working in the fields outside of academic and pharmaceutical institutions interested in profiting in this field as they see it as a mainstay. Next slide. So I would like to discuss about how pharmacognosy is currently facing three new challenges that have been applied before. The first challenge is the evaluation of medicinal drugs of ethnic groups, which are used in medicinal products and are based on cultural tradition in which the therapeutic system is not based on receptor theory. Principles of traditional Chinese medicine, traditional Indian medicine, and traditional Japanese medicine and Mongolian medicine. Coming to the second challenge is the evaluation of biogenetic material. So this challenge is mainly obtained from the extracts and pure substances which are obtained from medicinal plants. Its preparations do not have a legally binding character of the drug, but hence they are used as nutraceuticals, dietary supplements, or novel foods. Nutraceuticals are often used as a part of controlled therapy, sometimes even in self medication as the therapeutic agent itself. But nowadays, the situation is still complex and need to be addressed by legislation. The problem of nutraceuticals, even though they are used as foods, must be approached accordingly based upon the modern knowledge. And the third challenge is the pharmacognosy of microorganisms, and the use of these organisms was first mentioned 50 years ago, and its deeper development took place about 40 years ago in Sweden. Next slide. So, in order to assess the validity of pharmacognosy, it is necessary to name the areas which are included in the pharmacognosy either fully or to some extent. So, mainly I would like to discuss about four disciplines. First one is biological disciplines, chemical disciplines, production of raw materials, and technical disciplines. In the first biological discipline, there are various factors like pharmacognosy, which includes the basic verification methods, which involves the macroscopy and microscopy of the crude drugs. And the second factor is the pharmacobotany, which involves ethnobotany, physiology, and biochemistry of secondary metabolites. And third factor is genetics, which include gene recombinant technology. And the next factor is biotechnology, which includes microbiology, production of substances by various cell types, such as in vitro usage of recombinant DNA technologies and enzymes fixed to a carrier. And the last one is pharmacology and toxicology of natural substances, the basic characteristics of biological effect, the primary toxicity of newly isolated compounds, and detection of undesirable impurities. And coming to the second one, chemical disciplines, it mainly involves isolation of phytochemical procedures such as obtaining the active constituents of the drug. And the second factor is analytical phytochemistry, which involves both qualitative and quantitative analysis of chemistry of natural compounds. And the next factor includes preparative organic chemistry, that is, preparation of derivatives and structure of modification of isolated compounds. And the last factor is structural analysis methods. And the next one is production of raw materials. Production of biogenetic materials such as agrotechnics in the field production, technology of natural drugs, the processing of biogenetic raw material, and the substances which are used for isolation on an industrial scale. And the last one is technical disciplines. Technical disciplines include computer based methods such as chemical docking, cataloging, and classification of natural raw materials. The sum of all these disciplines, mostly which are preparatory, creates a whole set of form cognosy, and it is unimaginable to single out in any of them. Next slide. In this slide, I want to explain how we gain medicinal knowledge from the past. So, there is a precise date of acquiring knowledge about the use of medicinal plants and other natural substances is very uncertain. However, ancient tales, myths, and scriptures and other historical literature reflects the preliminary diagnostic approaches adopted for identification of medicinal substances and their medicinal properties. A general opinion is that primitive man might have started using these local herbs either to alleviate illness and injuries, either accidentally or based on superstitions, perhaps through survival instincts that they were curative agents for many diseases that afflicted them. This knowledge was passed down orally from generations to generations, and lack of documentation of such remedies makes it difficult to know how long a particular remedy has been used and whether the preparation or administration method has ever been altered or not. Unlike conventional medicines, which are only approved for public use after carefully planned laboratory research followed by field trials on animals, both for toxicology and effectiveness, these ethnomedicines or ethnoremedies depend only on the historical evidence of use as a proof of safety and effectiveness. However, there are some possible ways where humans have used or discovered and validated these ethnoremedies or ethnomedicines. The possible ways, which include the following, are during the gatherer-hunter stage, humans would have observed that ingestion of certain plant food material, which is obtained from either parts of the plant, such as roots, seeds, and fruits, was followed by some profound physiological changes, such as sedation, euphoria, hallucinations, diarrhea, vomitings, etc. He therefore learned to associate some pharmacological effects with such specific plants. Somewhere in Mexico, a native Indian who consumed god's flesh, which is known as Mexican mushroom, and the botanical name is Psilocybe mexicana, and experienced hallucinations. Then the man came to recognize that he could use his mushrooms before rituals whenever he wanted to communicate with God and through self medication by animals. During the gatherer hunter stage, and even much later, man observed that wild animals ate some plants and they exhibited abnormal behavior. For example, somewhere deep in the Congo rainforest, a native observed with amusement that wild boar digs up the roots of the plant Tabernath iboga and ate the roots to go into frenzy, jumping around and probably trying to run away from frightening visions. He immediately associated the plant with hallucinations. Yet another person in Abyssinian highlands observed that wild goats fed on the shoots of the plant Catha edulis were excessively stimulated. Out of curiosity, he chewed the shoots and it became hilarious. Next slide. Apart from the accidental discoveries which were referred in point 1 and point 2 above, there is evidence that man actively looked for medicines at the early stages of evaluation, making use of the doctrine of signatures. According to this doctrine, the physical characteristics of the plant, such as color, shape, form, and etc., and its productivity were indicative of possible medicinal values. Thus, the existence of plants as medicinal value came into existence. Now we could see some examples of the plants such as Ravolfia root, also known as Sarpagandha, which has snake repellent properties, was used in Ayurvedic medicine for snake mites because it is shaped like a snake. Another example, such as Chinopodium wormseed, was used as anti-alimentic, and saffron flowers were used for the treatment in jaundice, and plants with heart-shaped fruits were used for heart diseases, and plants with roots shaped like male organs were used for impotence problems. In addition, plants bear a lot of fruits are thought to have agents that promote fertility, and plants that exude milk-like substances are thought to improve the milk yield, and plants that produce red liquids are thought to enhance the blood, etc. Next slide. As a form of inheritance from grandparents or grandparents, evidence based on the use of treatment is the most widely used criteria to determine whether the treatment is safe, effective, or not. In most cases, ethno practitioners will try to find out how many people use the same medicine for treatment, either on animals or humans. The more users, the higher the credibility of treatment, and plants with pleasant fragrance were used to mask perspiration and flavor food. And they were also used to spice and preserve food. This is how some of the naturally occurring nutraceuticals were also discovered. And scientists like Christopher Columbus, Vasco da Gama, and many other ancient explorers made the journeys to find alternative routes to the land of spices. And we can also observe documented research findings. In the last few decades, there has been a rush to document traditional remedies. Researchers have been surprised to discover that many of the traditional health remedies used in Africa are also used in South America or Asia. Sometimes the plant remedy is used for different treatments, whether structured or conventional trials have been carried out or not on their effectiveness. These documented treatments can easily be accepted as alternatives to unavailable or high cost conventional treatments. Next slide. Modernization of pharmacognosy. As decades are passing by, we can see there are a lot of trends and modernization of pharmacognosy. Several historical indications have shown that pharmacognosy was evolved from early scientific studies on medicinal herbs, and it includes botanical and phytochemical aspects of medicinal substances. There is a considerable evidence that illustrates the vigorous contribution of European scholars in the documentation of traditional medicinal knowledge since the time of Hippocrates. However, available records show that the first attempt for institutionalization of pharmacognosy was developed in the field of research and teaching. Next slide. Coming to the treatment, so each and every herb has some medicinal property. That is how many herbs are used in for the treatment of many medicinal properties. So each herb has its own properties, which include its energy, flavor, movement, and its related meridians to which it is connected to. The four types of energies are cold, cool, warm, and hot. Usually, cold or cool herbs will treat fever, thirst, sore throat, and general heat diseases. Hot or warm herbs will treat condensation in the limbs, cold pain, and general cold diseases. Generally, we can see that five flavors of herbs are pungent, sour, sweet, salty, and bitter. Next slide. Next slide. So, pungent herbs are generally used to induce perspiration and promote circulation of both the blood. And sour herbs exert mainly three functions. They either constrict, obstruct, and solidify. These herbs are good to stop perspiration and diarrhea. Sweet herbs also exhibit three main functions like sour herbs. So, they exhibit nourishing, deficiency, and harmonizing. So they are mainly used to relieve pain and slow the progression of acute diseases. And salty herbs are used to soften hardness, lubricate intestines, and drain downward. And these salty herbs are mainly used to treat heart stools with constipation or heart swellings, as in diseases like goiter. Next, coming to bitter herbs, they are mainly used to induce the bowel movements, where bitter herbs are used to reduce the fevers and hot sensations, dry dampness, and nourish the kidneys. Next slide. After consuming these herbs, it undergoes absorption, and the herbs can move into four different directions. So, first direction is it moves upward towards the head and downward towards the lower extremities. It moves inward towards the digestive organs and outward towards the superficial regions of the body. And the third one is it moves up. The herbs move upwards and are used for falling symptoms like prolapsed organs. And the last one is downward moving herbs are used to push down surging symptoms like coughing and vomiting. Next slide. Next slide. So in this slide, I will be explaining about the challenges for developing standardized herbal drugs or formulations. So there are four basic principles which need to be addressed for the development of clinical use of herbal medicine. First one is there must be uniform regulation and standardization protocol for the product being studied or being used clinically. This is considered to be one of the major drawbacks in the case of herbal medicines as there is no standardized protocol in order to follow. And the second one is there must be adequate scientific data to claim the safety of the product intended for human use by rigorous clinical research. And the third challenge is there must be enormous scientific data produced or established by vigorous clinical research for claimed therapeutic effect to the patients. And the fourth one is the guidelines on quality evaluation and development of herbal medicine is utmost important in setting the pace of global acceptability. Next slide. And after facing all these challenges, it is also important to prepare the quality. It is also important to prepare the need of quality assurance. So quality assurance plays an important role in maintaining the quality and standardization of the herbal medicines. Hence they are essential for the monitoring of the entire process of formation of herbal drugs, bioactivity guided fractionation, and bioprospection, utilizing technical standards as per the authorized gazette or pharmacopoeias. The standardization as DIPT is a process of describing a whole set of inherent characteristics, fixed parameters, and definitive qualitative and quantitative values which assures the quality, efficacy, safety, and reproducibility of the herbal medicines. Next slide. So there are various factors which affect the quality of herbal drugs. The first factor is the complex nature of phytochemicals, the active principles, absence of wide range of major minor compounds, associated natural variability inherited with plants in wild and non-wild varieties, and shortage of standard fingerprints of phytochemicals to record or analyze efficacy within different batches. And the last factor is variability in spectrum of pharmacological potential of species which are grown naturally versus chemo varieties. And the other factor is sophisticated analytical techniques are constantly developed and upgraded day by day to isolate the compounds, identify the constituents, and quantify the constituents which are responsible for the therapeutic potential of herbs or plants. It is also evident that these metabolites vary qualitatively and quantitatively. And variability in quality of raw materials, the source of origin also plays an important role where the quality of the herbal drug gets affected. Next slide. And standardization of herbal drugs. So herbal drugs need to be standardized before going into the market. So it includes various factors to be determined, such as the taxonomic authentication of the plant material should be done, determination of foreign matter, determination of organoleptic evaluation, and it is also important to know the macroscopy and microscopy of the particular drug and to determine the volatile matter of the drug, ash values, extractive values, and chromatographic profile of the drug can be done by using various chromatographic techniques. It is also important to know the marker compounds and the pesticide residue of the drug, the microbial content of the drug, and heavy metal contamination should be determined. Next slide. Basic tools for quality evaluation of herbal drugs. So, what are the basic tools for the quality evaluation of herbal drugs? So, quality evaluation it starts from the proper identification of desired plants, collection of the right plant material, season of collection, extraction, and purification processes. So, while going for evaluation of herbal drugs, it is very important to keep all these factors in a mind as the identification of the plant, not only right from the identification of the plant, collection of the plant material at the right time, at the right season, its extraction procedure, and purification processes using various technical methods also plays an important role. In order to maintain proper quality evaluation of herbal drugs, regulatory authorities like Ministry of Ayush, Indian Council of Medical Research, and National Medicinal Plant Board have provided guidelines and important tests which are required for the development of quality assurance parameters for herbal drugs as mentioned. Next slide. So, challenges in herbal medicine. So, while preparing herbal medicines, there are some few challenges in herbal medicines which need to be considered. The first challenge is management with ranges of risk. It is always important to check the risk, how far we need to risk it for. And the second one is communication of uncertainty. So while we are collecting the plant material, it is important for us to communicate and regularly be there to know whether the drug is properly collected or not. And the third factor is the pharmacological, toxicological, and clinical documentation is very important. So all the pharmacological work, the toxicology, and the documentation of the work should be properly done. And the next factor is the pharmacovigilance and understanding why addition of harmful additive works. It is also important for us to know whether the additives are harmful and what is the role of additives in that particular herbal medicine play an important role. And the next one is evaluating the drug interactions. It is also important for us to know what type of interactions occur in the drugs when we use additives or when we mix with other drugs. And the next challenge is constraints with clinical trials and the people available. It is also important for us to go with the clinical trials in order to know whether the herbal medicine is safe for the usage or not. And the next challenge is standardization. So standardization, as I already told you in the before slide, it is very important to follow all the standardization methods. And the last one is finally, it is important for us to know the safety and the efficacy assessment of the herbal drug. Next slide. Evaluation of new herbal products. So, evaluation of herbal products also includes few characteristics to be followed. So, for new herbal products, mainly we see the characteristics of new substances, whether new substances are evolved or whether the existing substances are already proved. And the next evaluation is history and pattern of use, whether that particular herbal product has certain history and what is the pattern of use should be noticed. And the third one is any adverse reactions. So whether that particular herbal product is having any adverse reactions or not should also be checked. And the fourth one is biological action. By performing biological activities, we need to know whether what biological action that particular herbal product is used. And we need to also consider the toxicity and carcinogenity of the new herbal product. And finally, clinical trial data. Next slide. And what are the constraints in the herbal medicine? So, constraints are the limitations which are associated in handling of medicinal plants. So, coming to constraints, there are a lot of constraints to be considered while we are handling the herbal medicine. First one is indiscriminate harvesting and poor post-harvest treatment practices. Whenever we are going to herbal medicine, it is always important to follow certain standard procedures for the cultivation and collection process. So, if whether the harvesting techniques are properly followed or not, This leads to one of the disadvantages in preparing the herbal medicines. And the second one is lack of research on the development of high yielding varieties, domestication, etc. So there is a need of knowing the history of the plant, and we also need to do a lot of research, as we can see many people without, due to lack of knowledge, they cultivate the plants and they get low yielding varieties. And it is also important during cultivation to properly keep an eye such that no domestication when the crop is not affected by certain rodents. And the next constraint is poor agriculture and propagation methods. So it is always important for us to know the soil and soil fertility, various techniques, various factors play an important role in the cultivation of the plants. And it is important depending upon the choice of the plant to be cultivated. So certain propagation methods are to be followed. And the next constraint is inefficient processing techniques, which leads to low yields and poor quality products. So, processing techniques, we should have a proper knowledge on the processing techniques, which otherwise leads to low yield, and the quality of the product also affects. And the next constraint is pure quality control procedures. It is very important to follow quality control procedures in order to get a good yield of herbal medicines. And lack of current good manufacturing practices. Whenever we go for cultivation, it is important to follow good manufacturing practices. And this is considered to be one of the constraints in the herbal medicines. And the next constraint is lack of R&D on product and process development. So lack of research also on the product to be maintained and the process to be developed also is considered as one of the drawback. And difficulties in marketing. Even though cultivation and collection and post harvesting techniques are properly done, sometimes marketing also plays an important role, which is also considered to be one of the limitations. And the next one is lack of trained personnel and equipment. So during cultivation, it is important for us to keep trained personnel with good sound knowledge and the equipment which can be handled properly. And this is also considered to be one of the constraints. And the next one is lack of facilities to fabricate equipment locally. So look, even this is also playing a major role where where there is a where people are unable to keep the equipment properly due to lack of facilities. And the next one is lack of access to latest technologies, and these are the constraints which are associated with the dealing of herbal medicines. So, in addition, the processing of herbs, such as after cultivation, even the processing of herbs also plays an important role. So, if the herbs are not properly processed, such as heating or boiling, it may alter the dissolution rate, sometimes even the pharmacological activity of the organic constituents. Similarly, a host of environmental factors, which includes soil, altitude, seasonal variations in the temperatures, atmospheric humidity, length of the daylight, rainfall, pattern, shade, dew, and frost conditions also affect the level of components in any given batch of an herb. Other factors which includes infections, insects, plant intensity, competition with other plant species, seeding time, and genetic factors also play an important role. Plant collection for the use in botanicals is one of the factors of concern for quality. Plants collected in the wild may include non-targeted species, especially either by accidental substitution or intentional adulteration. Adulteration of herbal products can be made in various common ways, where adulteration is made by substituting with easily available or cheap plant species, and sometimes by spiking of the product with synthetic constituents. So during the when we are dealing with herbal medicines, adulteration is a major factor to be considered because of the because of the available marketing. There are many chances of adulteration, and we can see many products which are being adulterated. Next slide. Next, coming to innovations in novel drug discovery. So, what are the innovations in novel drug discovery when coming to pharmacognosy? The current challenges to the use of natural products and difficulty in accepting scientific evidence of natural products, as I previously mentioned, led to the development of some blockbuster conventional medicines. Searching for new drug candidates from natural products is often made difficult by the complexity of the molecular mixtures. So, as the plant contains several complex molecules, it is difficult for us to know which type of compound, whether a single compound or a mixture of compound is having the activity or not. And the next innovation is the therapeutic activity of plant extracts is usually because of the synergistic and simultaneous action of several chemicals. And the next one is plant-based drug discovery. Therefore, must start with a combinatorial approach when evaluating compounds. So it is always recommended for plant-based drugs to start with a combinatorial approach for evaluating the presence of active constituents of the drug. And the next one is the advent of novel technologies, which includes quantum computing, profiling techniques, computational biology techniques, big data, and artificial intelligence will enable scientists to use a combinatorial approach to harness the therapeutic properties of plant-based natural products and simultaneously study their molecular effects in physiological conditions. As recently we can see there are various novel technologies such as artificial intelligence, computational biotechnologies where that within less time new compounds are evolved. And the next one is innovative drug design from natural products is needed to combat global health challenges with the assistance of technical innovation. And the next innovation is ultimately more research should be focusing on combinatorial effects of chemicals from plant extracts and not just single compounds. Next slide. So, multidisciplinary approach to natural drug discovery using innovative technologies. So, recently there are multidisciplinary approaches in when coming to the discovery of natural products. So, the successful use of such an approach will allow the development of next generation drugs to combat the ever increasing health challenges of today and the future. So the first approach is genomics in plant-based natural products, which includes identification and biomarker identification, proteomics in natural product validation, and biomarker identification. And the next approach is metabolomics and metabonomics approach to natural product drug discovery. And the last one is big data in drug development for natural product drug development and precision medicine. Next slide. So, coming to the clear picture of genomics, which are used in plant based natural products, genomic methods are important in establishing an accurate identification method for plants and natural product species. This is an evolving technique which is for developed for techniques such as DNA barcoding, which are used as which are used to know the standard DNA regions for species level identification. This DNA barcoding utilizes genomics, which will provide a more robust and precise identification compared to traditional methods of morphological identification and local traditional names. So, presently, this genomics has gained a lot of importance rather than knowing the plant or identifying the plant based upon the morphological identification or by its traditional names. And the next one is proteomics in natural product validation and biomarker identification. Proteomics approaches to innovative drug discovery from natural products have the potential to elucidate the protein expression. So, it is mainly used for elucidating the protein expression, the protein function, metabolic and biosynthetic pathways, which are based on therapeutic effects, translating to consistency in quality and profile of the product. Proteomics application has been successfully used in identifying species of Chinese herbal medicine. An example of such proteomics, Penang's ginseng versus Penang's quinquefolium has been done, and it has given possible results in the biomarker identification. And proteomics is considered as an effective way to elucidate multi-target effects of complex natural product preparations as well as the discovery of multiple compounds and fractions, characterization of natural products, and ultimately a molecular diagnostic platform. Next slide. and metabolics and metabonics approach to the natural products drug discovery. How this metabolic profiling is important in the case of natural products. So, metabolic profiling is mainly used to identify and quantify the complete set of characteristic metabolites, while metabonomics broadly describes the evaluation of global and dynamic metabolic responses of the living systems either to biological stimuli or genetic manipulation. Metabolic and metabonomics profiling use in the mainly they are used by sophisticated equipment such as in mass spectroscopy, and ultra performance liquid chromatography. By using such sophisticated instruments, we can potentially elucidate the pharmacodynamic and pharmacokinetic properties as well as the toxicological value of the natural products can also be developed using metabolics and metabonomics. And the next one is big data in drug development for natural drug development and precision medicine. So coming to omics analysis, which includes genomics, transcriptomics, proteomics, metabolomics, and metabolomics, it results in generation of a complex multivariate dataset that requires computational and chemometric tools for interpretation. So recently, this has been an evolving technique where Interpretation of interpretation of the data is done by computational techniques. The use of these computational platforms such as bioinformatics and multivariate statistical tools will allow the application of omics multidata to elucidate the pathophysiological effects, target specificity, and molecular effects, as well as it elucidates the pharmacodynamic and pharmacokinetic and toxicological characterization of natural products and the compounds. Applications used during the drug discovery process, such as docking and virtual screening, can also be used as novel machine learning algorithms, such as deep learning. Machine learning methods can be used for virtual screening of thousands of compounds, which allows the utilization of data from high throughput screening. Next slide. So, what are the trends in pharmacognosy? By explaining all the above innovations in novel drug discovery, there are few trends in pharmacognosy which has become a multidisciplinary research, a high-tech science of natural medicines in terms of assessing their purity, potency, and consistency, especially in its methodology of using faster and more effective analytical methods, high throughput screening, target based drug discovery, and in silico methods for virtual ligand screening. Molecular pharmacognosy, genomic pharmacognosy, and metabolic pharmacognosy have been contemplated as the most promising approaches of pharmacognosy to meet the emergent trends in molecular biology, biotechnology, and analytical chemistry of natural medicines. For example, nowadays we can observe DNA barcoding represents an essential addition to the wide range of robust methodologies which is used to identify and authenticate the natural drugs and the pharmaceutical products. Challenges in pharmacognosy. So, as I already told you, reverse pharmacognosy together with reverse pharmacology, when they are combined, plays an important role in modern pharmacognosy. Reverse pharmacognosy allows us to identify whether the molecule from an organism are responsible for the biological activity and the biological pathway involved. An exciting outcome of this approach is that it not only provides evidence of the therapeutic properties of plants which are used in traditional medicines but also position other plants containing the same active constituents of the same usage, thus creating the curative arsenal. Through this we can develop new botanicals can be evolved by using reverse pharmacognosy technique. And this reverse pharmacognosy is organized into five components, which are three-dimensional structural database of natural compounds with its equivalent of natural compound library formatted from high thorough inputs, a target database, a virtual screening tooling, and an ethnopharmacological database. Next slide. And reverse pharmacology, also known as target based drug discovery, is based on two processes. So, the first process is it is hypothesized that modulation of the activity of a specific protein target will have beneficial therapeutic effects, and subsequently, studies in small molecule chemical libraries are used to identify compounds that have a high affinity with the target. The results of this screening are used as starting points for the discovery of drug candidates. This method has become popular for sequencing the human genomes, which enables rapid cloning and synthesis of large number of purified proteins and is currently the most widespread in the field of drug research. In contrast to classical pharmacology, the effect of in vivo identified active compounds also is predicted by the reverse method, which is only used in the final stages of drug discovery with a reverse pharmacological approach. It is also very important for the assessment of natural substances. Next slide. And I would like to, in this slide, I would like to explain what is pharmacognosy and what is reverse pharmacognosy. So, in the basic pharmacognosy, we study the medicinal plants and their extracts, constituents, and its phytochemistry. After that, it consists of the following steps. Firstly, selection of plants. So, plants are selected on the ethnopharmacological knowledge, the specific therapeutic area which is desired for the treatment. So, using knowledge from different cultures, it increases the probability and ability to identify effective materials that correspond to the therapeutic area. One important consideration is the availability for sufficient quantity of samples for testing and subsequent development. After the plant is selected, the next step we go through is extraction. And as we all know that extraction can be defined as a process of removal of soluble materials from an insoluble residue, either liquid or solid, by treatment with a liquid solvent. And this extraction is generally done by solvents of different polarity by using different extraction methods. Next slide. After extraction, the next step we follow in pharmacognosy is biological evaluation. So, biological evaluation is done to estimate the potency of the crude drug or its preparation, which is done by means of its effect on living organisms, either like bacteria, fungal growth, or entire animal, which is known as bioassay. So, bioassay guided fractions are responsible for screening of biological evaluation. And the next step we follow is characterization. After bioassay, we go for isolation of active compounds from the extract, which is considered to be an important step, which is done by chromatographic techniques. So the active compounds are isolated using chromatographic techniques like TLC, HPTLC, and HPLC. The main constituents of the drug are studied with the aid of HPTLC. So first, after getting the extract, we go for preparative TLC, and depending upon the fluorescence of compounds which are observed under UV light, we go for HPTLC studies, which are considered to be the modern method for the identification of the type of compound of the drug can be done by HPTLC studies. And the modern spectroscopic techniques also using HPLC coupled spectroscopic techniques such as HPLC UV, HPLC MS, as well as HPLC NMR. Next slide. What are the various parts of reverse pharmacognosy? Till now, we have seen the basic parts of pharmacognosy and what are to be followed. And in this slide, I'll be explaining about the various parts of reverse pharmacognosy. So, reverse pharmacognosy include structural database for natural compounds, target database, virtual screening tools, ethnopharmacological database, and traditional knowledge digital library. Next slide. Next slide. Structural database for natural compounds. So the natural compounds which appear in the published literature and the compounds which are found in the commercial database forms the structural database, which is also called the virtual chemical database. So the sources of these compounds are available and are frequently the method applied for the extraction is also described. So Concord is a VCDB contains more than 1 lakh natural compounds along with their 3D coordinates. Chemical diversity of the compound is the final criteria for the selection of compounds for virtual screening. And the next variant is target database. The target database is composed of 3D protein structures which are determined by X-ray crystallography or homology modeling. The majority of these structures are from humans, although it contains proteins from other sources such as viruses and bacteria. So the protein or the ligand pair interaction energy is obtained and it involves retrieval of active ligands from a set of 100 non-active compounds. Next slide. And virtual screening tools. The basic goal of virtual screening is the reduction of enormous virtual chemical space of small organic molecules to synthesize and screen against a specific target protein to a manageable number of compounds that exhibit the highest chance to lead a drug candidate. So there are two methods for virtual screening. So the first method is based on the ligand properties, where we consider as our physico-chemical properties, which is also known as one-dimensional data. And the second one is fragmental description, which is also known as two-dimensional data. So the two-dimensional data where the techniques of quantitative structural activity relationship and screening, which is based on the target properties, which requires knowledge on 3D structure of the target, and the ligand, which are techniques of de novo design and docking, which involves generating new ligands or adjusting ligands in the active site of the target respectively. And next is ethnopharmacological database. So in order to develop the botanical data, natural chemical structures, biological testing of extracts and compounds, this ethnopharmacological database has been developed. So in this ethnopharmacological database, the family, the genus, species, common names, synonyms of the plants are included in this database. So this database accelerates the discovery of bioactive ingredients. So bioactive ingredients examples such as anti-inflammatory compounds. And this database contains botanical information on plants and their traditional uses, its phytochemistry data associated with the biological activity of plants, and this database also acts as a link between plants, molecules, and activity. Next slide. The next one is traditional knowledge digital library. In order to prevent the grant of patents based on Indian traditional knowledge, Government of India has undertaken an ambitious project of creating a traditional knowledge digital library. Since many of the people are due to lack of knowledge on medicinal plants, hence the government has initiated this traditional knowledge digital library. This is a joint venture of Council of Scientific Research and Central Council for Research in Ayurveda and Siddha. This project is intended to cover about 35,000 formulations which are available in 14 classical texts of Ayurveda to convert the information into patient patent compatible format. The work has been initiated with cooperative setup of 30 Ayurveda experts, 5 information technology experts, and 2 patent examiners. The digital library will include all details in digital format about international patent classification, traditional research classification, Ayurveda terminology, concepts definitions, classical formulations, its doses, disease conditions, and references to documents. Next slide. So in this slide, I will be explaining the comparison of pharmacognosy and reverse pharmacognosy. So what is basic pharmacognosy and how reverse pharmacognosy plays an important role. So in pharmacognosy, firstly, we obtain plants from the vegetative realm, and these plants are subjected to the preparation of extracts, and these extracts we perform the biological active tests, and from this biological activity we go for bio-guided identification in order to know the presence of bioactive molecules which are responsible for its medicinal properties. Whereas in reverse pharmacognosy, the natural compounds are taken from the molecular database or the molecular library which is available, and then it is subjected to virtual screening tests or biological tests, and then knowledge based identification from that particular plant. We can observe that in pharmacognosy, in the first step itself, we are taking plants. In reverse pharmacognosy, the last step we are considering is the knowledge based identification of plants. Next slide. So this is a diagram representing the origin of network pharmacology from pharmacognosy. If you see the pictorial representation in the year 1999, the traditional medicinal symptoms are related to molecular work, and then slowly in the year 2002, network regulating effect of traditional medicine prescriptions has started, and progressing slowly to the year 2007, biological network of Hort syndrome and Paul syndrome based on plants is determined. And in the year 2008, network pharmacology came into existence, which is considered as the next generation medicine research model. And in the year 2009, herb network and biological network and phenotype network came into existence. In the year 2011, network target network pharmacology research using traditional medicines has been developed. Next slide. And this is a slide representing the herbal drug opportunities in the 21st century. We can see from ancient knowledge, the bioresources is developed into modern science, which includes herbal medicine, which is converted to phytopharmaceuticals, nutraceuticals, and cosmeceuticals, which are useful for both health, economy, and employment. Next slide. Next, coming to conclusion, I would like to conclude by telling that India has moved forward in advocating the global usefulness of Ayurveda, the scenario of healthcare through global networks. As a result, many foreign countries have been looking at India for understanding Ayurveda and incorporating it to education, research, and practice to meet the overwhelming desire of consumers to access complementary and alternative medicines. The role in challenging the information of Ayurveda and opening up new opportunities for the spread of Indian medicine into foreign institutions and general public awareness is building about Ayurveda has been identified as an important thrust issue. So, in such an arena, reverse pharmacognosy, by making use of virtual screening tool and important databases, can play an important role in reducing the time and wealth. It can accelerate the drug discovery to a considerable extent. Ma'am, I'm unable to hear you.
Yes. Thank you, ma'am. It was a wonderful session. I request all the participants, if you have any queries, please post in the comment section. Yes, ma'am, we have a query. Can you please explain the scope of quantum computing as a novel technology in pharmacognosy?
Yes, sir, because this is considered to be as one of the important novel technology because through database in pharmacognosy we do through morphological identification we start the research work. But through quantum computing, everybody can know, everybody can have a knowledge on computing based upon the high technologies used. So depending upon the quantum, we can select the plant and we can proceed for research. Nowadays, this database is helping us in finding out the medicinal plants rather than only pharmacognosys depending upon the medicinal plants, depending upon the morphological characteristics and the taxonomy of the plant.
Yes, ma'am. Another query. Why addition of harmful additives work? Challenges in herbal medicine.
Yeah, it is important for us to know there are harmful additives as well as useful additives. While we are preparing herbal medicine, it is important to know there are some types of food interactions. Herb interactions do occur, herb drug interactions do occur. So we should have a thorough knowledge when we are using herbal medicine. What are the ingredients used? Whether there are possibility of additives, it is necessary to find out.
Ma'am, another query. Please explain the methods followed and processes of standardization of herbal materials.
Yes, sir. I already explained the standardization of herbal materials in my slide. As standardization of herbal medicines is very important, and this is considered to be one of the major effect in preparation of herbal materials. So there are different standardization procedures such as determination of heavy metals, determination of pesticide residues, determination of extractive values, determination of acid values. It is important to go for all the standardization procedures before marketing the drug into the public.
Any more queries from the participants? Yes, thank you, ma'am. I think there are no more queries in the session. I request Dr. D. Nivedita, ma'am, to proceed with the next session.
Thank you, ma'am. It has been a great honor to be a part of today's session on behalf of the staff and organization. I would like to extend my heartfelt gratitude to our esteemed guest speaker, Dr. Kumari Vedapriyama, Associate Professor of Adya College of Pharmacy. Thanks a lot for sharing your knowledge with us, ma'am. I would also like to thank our organization for your support in organizing the FDP, and my heartfelt thanks to the principal, Dr. K. Achyut Kumar sir, for always being a backbone and guiding us. Finally, I would like to thank all of you present here for making that time to be with us today and helping us make today's session fruitful. Thank you. Thank you, everyone.
Thank you, ma'am. I would like to thank Dr. Achyut Kumar, sir, convener of this FDP, and my dear friend K. Swati Priya, co-convener of this FDP, for recognizing the potential in me and finding and selecting me as one of the speaker for today's FDP. I thank the management of Srinivasrao College of Pharmacy for giving me this opportunity. Thank you. Ma'am, can I leave?